Alright, ladies and gentlemen, welcome back. We got one final category of macromolecules to talk about before we're done with this section, and that's nucleic acids. So, I'm going to give you mostly structural stuff here. Not going to spend crazy amounts of time on your nucleic acids right now beyond just its basic chemistry, because we're going to spend quite a bit of time come this spring talking about nucleic acids, what they do, how they function, how we make more of it. So there's more to come on this later this year. So in terms of types of nucleic acids, we're talking DNA, RNA, as well as this modified type of nucleotide called ATP. Okay. So in terms of function, well, your DNA's job really is to basically be that storage of hereditary or genetic information, and maybe that's not a terrible side note to add to yourself here. So DNA's main job is to be that storage molecule for all that information that ultimately is going to make you look and function the way that you do on a regular basis. Your RNA molecules, their job lands pretty much here in this second bullet, and they're going to assist with the production of proteins. So their job really is to carry the instructions from your DNA out into your cytoplasm where we actually build those proteins. And so they're going to get involved there as RNA molecules. And then the final function here, the storage and transfer of energy, that's ATP. Okay, so DNA is the first one, RNA is the second, and ATP is that third function. So in terms of chemical structure, what do I need to know? Well, carbon, hydrogen, oxygen, again, nothing's changed. These guys also have nitrogen like your proteins do, but now they've got the phosphorus instead of the sulfur. So they contain phosphate functional groups. If we take a look, let's take a look at ATP first. This is a modified nucleotide, so we'll talk nucleotides are your building blocks in just a second. And I say it's a modified nucleotide, and you'll see what I mean in a moment. What I'm looking at is, here's the basic structure for ATP. So I've got this ribose sugar, I've got an adenine, which is a nitrogenous base, and then I've got 3 phosphate groups that connect here. Alright, and these 3 phosphates are really where I'm going to be able to store a lot of energy in the bonds that are holding these guys together. When we break those phosphates off, we release a bunch of energy, and then that energy then can get transferred when I transfer those phosphate groups to other molecules so that we can actually get those other molecules to do some sort of function in our cells. We'll spend a lot of time talking about ATP when we get to cell transport and cellular metabolism later this year. For now, just recognize the basic structure, know that it's a nucleic acid, know that it's there for storage of energy, and basic parts, and that'll do for now. We'll get into more of how this works a little later this year. So I said ATP is a modified nucleotide. Why? Well, nucleotides are your monomers or your building blocks for nucleic acids, and they've got 3 basic parts. They've got a sugar, and if I'm talking RNA, then I'm talking ribose, just like we saw in ATP. I've got a nitrogenous base. Alright, and we're going to see some different options for those bases in just a second. The adenine you just saw in ATP is a base. And then I also have a phosphate group. Okay, so ATP has 3 of those phosphate groups, hence the term a modified nucleotide. If we take a look, every nucleic acid has a pentose sugar, so I showed you riboses here. That's what's going on in your RNA. DNA has deoxyribose, and check it out. In terms of chemical structure, the only difference between RNA and DNA in terms of their sugar structure is ribose has an OH group coming off this second carbon. And guess what? Deoxyribose is deoxygenated. It's missing that oxygen, so it only has a hydrogen here, which is kind of interesting, and that's where the name comes from. Nitrogenous bases, we've also got some options going on here in terms of nucleotides. For DNA, I've got 4 different types of bases that I can see or I can find in that particular molecule, so 4 different types of nucleotides going on. And I can either have adenine, thymine, cytosine, or guanine. When I get over to RNA molecules, they've got the A, they've got the T. They don't have the T, instead they've got a uracil or a U, and then they also have the C and G. So both A, C, and G are found in RNA. Along with the uracil, the thymine is not in the RNA, but instead is over here in DNA. So that's the only major difference in terms of the bases that are present. So in terms of basic structure, if I were to break this apart, here's your DNA double helix that hopefully you're at least familiar with that basic shape. If I take a single strand of DNA, alright, we're going to talk about that being a polynucleotide in just a second. Right, it's your polymer. Zoom in even further, and here's your basic nucleotide structure: phosphate, sugar, and a base. So polymer names, alright, here I'm talking about polynucleotides if we're going to keep that naming convention consistent, right? We had monosaccharides, polysaccharides, amino acids, polypeptides, nucleic acids or nucleotides, and polynucleotides. And your 2 major examples of polynucleotides, or these big strings of nucleotides stuck together, are DNA and RNA. So some basic background in terms of DNA structure. Well, DNA is double helix, so we've got 2 strands that are held together. You got hydrogen bonding going on here in the middle between your bases. Okay. And those bases will only come together in certain patterns. So your adenine always pairs with a thymine, A with T, and cytosine always pairs with a guanine, so C with G, base pair rules, A with T, C with G. If you don't already know those, start learning them now because you're definitely going to have to be able to memorize those guys later. So I've got hydrogen bonding going on between those bases. That's what's going to hold my 2 strands together. But in the backbone of your DNA molecule are your sugars and your phosphates, so these alternate in this backbone. And we kind of take a look at DNA like a double helix or a twisted ladder. So think of your base pairs and the hydrogen bonding here being the rungs of those ladders where you put your hands and feet. And then your phosphate and sugars are the sidebars that you might hang on to. So this one's actually got 2 bonds you gotta know. So the hydrogen bonds that are holding your bases together, but then you've also got these phosphodiester bonds that are holding the phosphate from one nucleotide, which is right here, to the sugar of the next nucleotide above it. Alright, so phosphodiester bonds are what's going to hold those sugars to those phosphates and keep the sides of your ladder together. Your other polynucleotide is your RNA. Single-stranded, when we actually build this, we use complementary base pairing, even though in its actual functional form there's only a single strand. So, once again, kind of learn some of your base pair rules here. So C is still going to go with G when we're building this, but now it's A and U that pair together, because remember there's no thymine in RNA, it's uracil instead. Still have a sugar and phosphate backbone. Alright, and in fact they still have that phosphodiester bond right here. That might not be the worst thing to label. Alright, so phosphodiester bond right here between the phosphate of one and your sugar of the next.